Hello everybody, today we are going to solve the paper 2 multiple choice examination question paper variant 2 paper 2 October-November 2025. So let us start. And the first question is telling you the photographs show two different rats and it's telling you which statement about these rats are correct. So let's first consider the names. It's telling you in the first name is Rattus norvegicus and number 2 is Rattus rattus. So basically the answer will be A because a genus it's always coming the first name. In the binomial nomenclature, as you can find here in our syllabus, that the genus is always the first name. So looking in the first name, we can find that this is Rattus and the other one is also Rattus. So basically both will belong to the same genus. Now not the same species because in the first picture of the first mouse it's norvegicus and number 2 it's Rattus, so they are totally different names for the species, so I cannot consider them as being the same. Question number 2, you can easily find it by just looking into figure number 2. Well, it's a bacteria and we know that the bacteria it's belonging to the group of the prokaryotes according to the classification that you had learned in chapter 1. So looking in here in cell 2, okay, first of all in A it's a protocotist, which is also, it will be wrong. C it is wrong and D will be wrong. So only the answer for bacteria, which is figure number 2 in question number 2, it is prokaryote, so the answer will be B. Now moving on to the next question, the diagram shows a plant cell. A biologist wants to find the number of chromosomes it contains. Which labeled part should be examined more closely? So basically you're going to examine the nucleus, and as you can see here in the diagram on the right hand side in here, as you can see in here that I have my cell, okay, and that cell contains what contains is a nucleus. That cell contains a nucleus and the nucleus contains the chromosomes inside it, and these chromosomes contains the DNA and the DNA finally contains the genes. So basically if in order for me to find this what I want in this question, basically I'm going to refer to the nucleus, and I left this picture for you guys just to understand it and memorize it as well. Which term describes each structure? Basically in here you're gonna just remember the definitions that you had learned before when you were learning the syllabus. So in brain it's an organ. Why it's an organ? Because it consists of different tissues which are working together to perform a specific function. And liver it's also an organ because it consists of many tissues, and one of these tissues is a muscular tissue. And neuron of course it's a cell because it's a nerve cell, okay, and it's found in our nervous system in the brain. Now moving on to question number 5. Question number 5 is basically very easy. Just memorize this trend and you will be good to go. A will be pairing always with T. So adenine, thymine, cytosine, guanine. Adenine will pair with double bond with thymine, and cytosine will pair with triple bond with guanine. So cytosine and guanine will be paired together by double bonds, and I had written for you here so that you can just understand it better. The A always come with it a T, the C comes with a G, the T comes with a A, the T comes with a comes with a T, G with a C, A with a T, T with a A. So just referring now to our answer in here, TGA, TGA, ATC, ATC, and TA. So it is already now compatible together. Number 6, which substance is an enzyme? Well, an enzyme will be pepsin. Why not fibrinogen? Because fibrinogen it was already fibrin, and it's in the cut wounds and cut bounds and heals of the skin, so it's already not gonna be the same as what we want. mRNA is the messenger mRNA. Ribonucleic acid. So basically ribonucleic acid in here I don't refer it as an enzyme because I'm gonna use it in protein synthesis. If you just refer to the protein synthesis lesson and DNA, you're gonna find it there. Now substance which is an enzyme, of course the only final answer will be pepsin, and another enzyme for your knowledge will be trypsin in the duodenum, as the pepsin works in the stomach and acidic pH, but duodenum works in the trypsin works in the duodenum at an alkaline pH. Why not saliva? Because saliva is the one which is it's a medium containing the amylase enzyme which digests the starch into maltose. You can find that in human nutrition lesson or animal nutrition. Number 7, which function is performed in the duodenum? So in the duodenum digestion occurs because in A assimilation occurs in the small intestine, which is not the case in this question. Egestion occurs through the anus, and ingestion occurs through the mouth. You're eating your food through your mouth, not through your duodenum at all. So basically the answer will be B. Now question number 8 is telling you here, the graph shows the changes in pH in the oesophagus, stomach, and duodenum. It's telling you here, now in the duodenum, exactly in the duodenum, what caused the change in the pH from acidic to be raised to an alkaline level? Why? What was released at that moment? Well, it's bile. Bile is released from the liver to emulsify the fats from larger droplets of fats into smaller droplets of fats. So gastric juice would be impossible to be used there, because basically gastric juice is acidic and it contains what? It contains pH at 2. And glucagon, it's an enzyme which will never, it's an amino hormone which is released by pancreas, which will never be alkaline at all. And trypsin, yeah, it is an enzyme, but that enzyme does not cause the alkaline pH. It only works in the alkaline pH. Check out for this difference. It is really very critical. Question number 9, which shows a small molecule right now here, and where that molecule is absorbed in the digestive system. Okay, so name of the molecule will be here water, and small intestine yes, and colon yes. Why A, B, and C are not correct? Basically because C, it's a disaccharide. It's not a small molecule at all. It's a disaccharide. It's 2 glucose molecules combining together producing your maltose, which is not the case in our question. So C just will be discarded at all. And when you reach A2 level, you're gonna find that maltose is also made from the other different compounds, but not our level now. Now, letter A here, amino acid. Small intestine, why not be absorbed in the small intestine? It should be absorbed in the small intestine because amino acid, it's a small molecule, it's a tiny molecule, and it should be absorbed in your small intestine. Glucose is the same goes, it's a monosaccharide and it's a small molecule, it's a tiny molecule, and it should be absorbed also in the small intestine. So basically, A and B should be absorbed, and they are not in this question, so they are not the answers. So just we're gonna discard them at all, and my final answer will be here D. Now moving question number 10. Question number 10 is very easy. Sitting you here that he put this plant into that blue dye, and basically, of course, any plant that you place in water, the water will be driven up by the cohesion and adhesion forces in the xylem vessels, so that they can reach what? They can reach the leaves, right? So in order for them to reach our leaves, you need the water to move through the xylem vessels. So for that to occur, you need a xylem vessel. So basically the question is telling you, these blue circles in right down here, the cut sections of the ciliary stalk, what do they represent? Of course, these positions of the xylem vessel. Why not phloem? Because phloem transport your sucrose by translocation and amino acid, which is not the case, because as we know that xylem only transports water and mineral ions. So basically it's a very basic idea. You have seen it in paper 6 before, in paper 4, and it's like very common in your syllabus. Now question number 11. Which process removes water from the surface of the spongy mesophyll cells into the air spaces during transpiration? Well, during transpiration we know that evaporation. I mean, we can link it together by a mnemonic, which is really very easy to understand. Now see, transpiration, okay, and what? Evaporation. We'll never forget it by this way. Check out, the common is here, Asian. Just memorize it by this way and you'll be very good to go. Like, you will never forget it by this mnemonic. I used to do this and it like really benefited me. So now basically evaporation, it's a passive process, and even if you don't know that information, just by your common sense, active transport will not be working because it requires ATP, and you don't offer ATP in plants in such a position. And B, circulation, it requires your blood circulatory system. And for C, evaporation, of course it is applicable because water will evaporate, converted to water vapor, and will move out of your leaves into the surrounding air. Osmosis requires a partially permeable membrane, so it should not be the case right in here. Okay, now question number 12. Question number 12 seems a bit tricky, but it's not tricky at all. It's telling you here the mass of water lost from a plant was investigated. The leaves of plant were covered with a type of grease that acts as a waterproof barrier. The environmental conditions remained the same throughout the experiment. The table shows the results of the investigation. Now just let us read the table word by word. First of all, in your x-axis on your first independent variable is giving you a treatment. No grease applied, and a grease applied only to the upper surface and to both surfaces of the leaf. And still you hear mass lost in 7 days, and still you hear the mean daily rate of water loss. Mean daily rate of water loss. Is not asking you for the mean daily mass loss or rate of mass loss. No, it's not asking you for that. He's asking you for a different complete thing. So basically what the students will do will take 8.7 divided by 7 days and will get B, which will be the incorrect option. But in order for us to solve it right, we're gonna get that 12.0, we're gonna subtract it. From 8.7, as you can see here. We're gonna subtract the 12.0 from 8.7, giving you a 3.3, okay? And now that 3.3 will be divided by 7 days, giving you 0.47148, which is gonna be rounded to two significant figures after the zero. So you're gonna count 4, 7, and here you go. The answer will be letter A, and it will be gram per day. So basically, this is a question you need to understand, grasp the idea. Because basically the question is just not that simple that he will give you 8.7 and you're gonna divide it by 7 and just get the final answer. No, it's not like that at all. Just need to read the question properly for you to understand it. Now, what is translocation? Basically, you just stated in the question of the xylem vessels. Translocation, it is the movement of sucrose and amino acid from the source to the sink. You tell me what is a source and what is a sink. A source is the leaves where it carry out photosynthesis, and a sink is the roots, the fruits, like for example the flowers, for example if I'm going to grow a strawberry plant, the strawberry will be the sink. So basically the answer will be here letter C. Now question number 14. Which row names the blood vessels? Be careful. Containing the highest blood urea concentration and the blood vessel containing the lowest blood urea concentration. And you're thinking that this is it's a tricky question. No, if you studied well, very easy. Basically the highest blood urea concentration, it is the hepatic vein, and that hepatic vein is just flowing containing what? Deoxygenated blood. And lowest blood urea concentration will be what? Will be the renal vein. Okay, so you will tell me hepatic vein and renal vein. You just go and memorize your circulatory system, and you will find that there is hepatic artery, hepatic vein, renal artery, and renal vein. Okay, just memorize these and you'll be good to go. And a very easy question, just nothing to worry about. Question 15 is telling you in here, the diagram shows the human digestive system. Where do cholera bacteria cause the secretion of chloride ions? Basically here in that question, the answer is letter D. You cannot tell me anus, because anus does not occur, it's only for digestion. A, it contains your food pipe where food just enter your stomach. And B, it's your stomach containing gastric juice at acidic pH. So it's a very easy question. Of course, the final answer and the only answer possible is letter D. And I just drew here an illustration for you. And if you check the previous video for May-June 2025 variant 1, I'm gonna leave you a link down. It was having the same question but in another way, but it contains what? It contains your gut. It contains your gut, which are these small intestine, and basically the water will flow out. The water will flow, what will flow out of these runways, okay, the small intestine into your blood, and basically we're gonna lose the water, causing dehydration. Question 16. Which row describes what happens when breathing in? Just focus with me. It's telling you what? Breathing in. So in order for us to just imagine this, I'm gonna leave you at the end of a page two pictures of how the diaphragm behaves when breathing in and breathing out. For this question, of course, is something to memorize. Diaphragm will contract and external intercostal muscles will contract. Be careful, intercostal muscles will be a little bit tricky because we have internal intercostal muscles and external intercostal muscles. These two things are totally different from each other. So just check page number 16 and it's telling you breathing in, okay? So in here in breathing in, as you can see here in breathing in, the external intercostal muscles contract and the diaphragm contracts and flattens. Diaphragm contracts and what? And flattens. And just let us go to our question in here. Yeah, the diagram here. The diaphragm contracts and the external intercostal muscles contract. So it's basically something you need to memorize and just get it, and it's a very free mark question. Now, a person exercised vigorously. Like just reading the first three words, I understood it's lactic acid and these statements and this story. For 1 minute and then rested for 1 hour. Which statements about lactic acids are correct? Lactic acid. During exercise, lactic acid is produced in the muscles and diffuses into the blood, which is already like right. Now, the blood transports lactic acid to the liver, for a breakdown by aerobic respiration using oxygen, and that is like where reoxygenation occurs. And now it's telling you here lactic acid is broken down by, oh no, no, no at all. This is already like denied and ignored. This answer is really very bad. It's telling you how anaerobic, and just we are delivering the lactic acid to the liver to be broken down by oxygen in an anaerobic way. Using what? Using oxygen. So 1 and 2 will be only the correct statements. So basically question number letter A will be wrong, C will be wrong, and D, of course, it's not only 2 because 1 is already right. Because lactic acid, after it's produced in the muscles, it will be delivered into your blood to flow to the liver to be easily broken down by the liver in aerobic respiration using what? Using oxygen. So just let us move to the next question, question number 18. Which letter identifies a synapse? We know that a synapse is a junction between two neurons. So basically, as you can see here, in letter B, we have here what? We have our sensory receptors. In letter A, we have a sensory neuron, and the evidence is that we have a cell body in the middle of the whole body. Okay? And a synapse, as you can see, I'm gonna maximize it for you. As you can see, it's a very small junction. It's like a space. We have a space here, and it's a junction right in here. So basically, the final answer and the only answer will be for that question is letter D. It's really basic. You just need to memorize it and just understand it. It's very easy. Okay. So the final answer here will be what? Will be letter D. Now moving on to the next question. Question 19. Person looks at a distant object in a dark area. I just want to highlight dark area. And the person then moves to a bright area and looks at a near object. Now we have two things that are moving in parallel ways together. We are considering our radial and radial muscles, and we are considering also the ciliary muscles and suspensory ligaments. So basically you just need to memorize it. It's very, very easy. Basically, you are moving now to a bright area. Now let us draw a diagram of the eye. Let you understand it now. And you are moving into what? Into a bright room. So in order for your eye to protect you from that high brightness of the lamp or whatever, like a sun, the radial muscles will relax, causing what? Causing the circular muscles to contract, narrowing the pupil of the eye. So the pupil constricts, so the pupil narrows. So basically, if you even don't know the concept of the ciliary muscles or suspensory ligaments, or you even does not bother about it, just just imagine it. Now he's telling you radial muscles of the iris relax. Okay, meaning that the circular muscles will contract. So the narrower the pupil will will get narrow. Basically means that the letter D will be wrong, because what? He's telling you here is the radial muscles of the iris relax, which is not the case. Okay, it's a case. This letter C and D are matching this part only. But starting from here, it's starting to get mixed wrong. It's telling you pupil dilates. How it pupil dilates? If the radial relaxes, now the circular muscles will contract. So contracting meaning that the pupil gets narrower. And I'm just gonna draw it for you here for you to understand it and imagine it as well. Now, let's say that I have here my eye. That's circular. Okay? And I have here my circular muscles. Circular muscles are in here. Circular muscles are in here. Circular muscles are in here. And I have my eye. I'll be like, make it here in blue. That pupil, okay? And now I have also my radial muscles. I'll be in yellow. So there's radial muscles, radial muscles, radial muscles, radial muscles. Here, here, and there. Now that eye, if it moved to a bright area, what? Let me focus with you again. Bright area. If I say bright area, meaning that my eye wants to get protected, I don't want my eyes to get burned in the sun or in the lamp or whatsoever. So basically, the circular muscles will contract, meaning what? Meaning that the circular muscles will contract, meaning that they will get closer to each other, closer, 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 causing what? Causing your eye, causing that blue area to be what? To be very, very little, causing that pupil to be what? To be very, very narrow in shape. So it will be very small like that. This is just an illustration for you to grasp the content, just not like a real diagram or whatsoever. If you want the real diagram, you can just check it on Google, and also it's found in Save My Exams. You just go check it there, and actually it's something to memorize. So if you memorize it, you're good to go, and it's very easy for you. I'll just leave it here for you guys to just use it. And yeah, moving question number 20. It's telling you here, which effect does adrenaline have on blood glucose concentration and pulse rate? Basically, adrenaline will be released just to increase your heart rate, and to increase your heart rate and to increase your blood glucose concentration, to increase the respiration, so that you will have more ATP produced by what? Produced by respiration. So basically here 20 will be D's answer, here will be what? Will be D. So pulse rate will increase so that the blood glucose and the glucose can reach to every part of the body for you to carry out respiration, releasing energy. Now question number 21. The diagram shows an experiment using two wheat shoe tips, X and Y. Which statement is supported by the evidence provided by this experiment? Basically, it's telling you here in X and Y, okay? So X basically will grow above, Y above, because oxygen is made in the shoot tip. You will tell me why oxygen moves through the plant by osmosis, because osmosis is just fluid. You need like water to move by partially permeable membrane. Oxygen is unequally distributed. No, oxygen is equally distributed, and the evidence for this is found in your notes, of course. And in May-June 2025 paper 4 variant 1, the last question was a filling blanks question, and it told you that the oxygen was equally distributed. So basically just go check that paper and you find that information is there. So basically A will be incorrect, C will be incorrect, and D of course will be incorrect. Oxygen does not inhibit cell elongation, oxygen stimulates cell elongation, and it was also found in the paper 4 variant 1. So just go check it from there. It's very really easy question, nothing at all. And even if you just don't like, you want to get familiarizing with this diagram, just use your knowledge, common sense, you get the answer. Let's letter B. 2022. Which action would help to limit the development of antibiotic resistance in bacteria? So it's telling you here, use bacteria only to treat bacterial infection, because as we know, viral infection, which is the viruses can only be treated by vaccination programs to develop what we know as herd immunity. But in this question, we need only to use antibiotic to treat only one thing. We don't our body like just to get that antibiotic as a common thing, like, well, hello antibiotic, how are you? Now you are common for me, I'm gonna get you, you'll not enter, you'll not cause effect in me again. But we don't want this to occur. So antibiotic will only be used in what? Into treating bacterial infection. Now I'm telling you here, what is an example of asexual reproduction? Asexual reproduction meaning that a single bacteria, it is dividing to produce two genetically identical bacteria. You will tell me why not letter B, because normal giving birth is basically sexual reproduction, because it requires two mates. A female bird laying eggs, 6 eggs, which will hatch into the same species, it is not a sexual reproduction, because of course they will not be genetically identical. They yes be the same species, but also you need to remember that female bird or a bird use sexual reproduction. Tomato plant producing fruits which may contain seeds, no at all, because in here we use cross-pollination and not the fact of asexual or sexual reproduction at all. Okay, I mean self-pollination or cross-pollination, use these. Now the question had came, diagram shows parts of two different plants, 1 and 2 of the same species. Okay, the transfer of pollen between the flowers between these two plants shown by the arrows labeled B and Q is telling you now which rows show cross-pollination. As we know that just by the definition, cross-pollination is transfer from a pollen grain from the anther of one plant to the stigma of another different plant. So 1 is a plant and 2 it's another different plant. So B, it is the only one for cross-pollination. Now the transfer of pollen grains that produces the most variation. Of course, cross-pollination produces more variation than self-pollination. It's just like knowledge, common sense for you to understand. Okay, why not Q is a cross-pollination? Because it is not a different plant, yes, yet it is a different stigma, but not a different plant. It is not a totally different plant. So just moving on to the next question. Question 25. Which substance is a hormone that is involved in the development and regulation of male secondary sexual characteristics? It is testosterone, and you cannot say insulin, because insulin, if your blood glucose is high, glucagon if your blood glucose concentration is low. Lactase, it is not at all, because lactase just digests lactose, and it's not the case in this question. And male secondary sexual characteristics, it's just a very small and very narrow topic. Male is testosterone, female is estrogen, progesterone, LH hormone, FSH hormone, follicle stimulating hormone. That's it. 26. Which statement describes the net direction of movement of a substance across the placenta? So urea moves from the fetus blood to the mother's blood. How? Because basically the fetus blood will have excretory products that will move to your own mother blood, because you don't have a way to excrete that waste products. Now, it's telling you here, carbon dioxide moves from the mother's blood to the fetus blood. Upon the conclusion I had just said seconds ago, A would be incorrect. Glucose moves from the fetus blood to the mother's blood. How? The mother should provide the fetus with food, so basically B will be incorrect, just common sense. Glycogen moves from the mother's blood to the fetus blood. Okay, the fact that it moves from the mother's to the fetus, which is food, is right, but glycogen is not right. We need a very small molecule, tiny molecule, a monosaccharide at least. At all, a monosaccharide only will pass. So glycogen is very big, is very huge. You cannot pass through from the mother's blood to the fetus blood. It's impossible. Okay. So now moving on to question number 27. Which label identified the uterus? Of course, it's letter C. Just something to memorize, because D is a vagina, A is the egg, is the ovary, and B is the oviduct where fertilization occurs. So it's not at all letter A or B or D. It's only letter C. Which method can transmit HIV? HIV, of course, it's sharing a needle with a person infected with HIV, because hugging a person infected with HIV, it is not it's not transmissible by hugging a person. Of course, if you hug a person with HIV, you will not be infected at all. It's only transmissible through your blood, or when you had a contact during sexual intercourse, which is only ways you can have HIV. But others, you will not have HIV at all. So basically, the answer here will be letter D. Now, which chromosomes can be found in a single sperm cell, X or Y? You will tell me, maybe some students will choose it X and Y. You just need to imagine that a female is giving you an X chromosome and a male is giving you a Y chromosome. Or the other case around, a female is giving you an X chromosome and the sperm cell is giving the female a Y chromosome. So it's either X or either Y. Okay, just need to keep that content in your mind. Question 30. Why are lymphocytes the only cells that produce antibody proteins? Well, antibody proteins, because the genes for antibodies are only expressed in lymphocyte. Basically, if I am having a cell and that cell contains genes, and these genes are only programmed, and they are just working, and they are just coded to produce one single product, which is antibody proteins. For example, if I have a 3D printer, okay, or I have a washing machine, and I just program that washing machine to just wash nylon clothes, don't wash cotton clothes. Of course, it will only wash nylon clothes, neither washing cotton clothes. So just memorize it like this. Just have the concept like this, understand it like this way. Of course, you cannot say that lymphocytes express all the genes. It will be incorrect. Lymphocytes have twice as many genes as other cells. No, all cells in the human body share equal amount of genes. Remember this. Other cells do not have the genes for antibodies. Of course, all of the cells have the genes for everything in your body, but the fact and the idea behind it is that it is expressed only in the correct time, in the correct place, and that concept you will learn in A2, not now. But just you have already a concept of it in the syllabus content of O level. Yes, it's present there, but it's not just more elaborated. Okay, it's just not elaborated that much. Okay. Question 31. During protein synthesis, what is the function of the ribosome? Okay, what is the function of the ribosome? Well, it assembles amino acids in a chain by using protein synthesis. Now, letter B, it carries a copy of a gene to the cytoplasm? No, because that one is done by an mRNA. It's not this. It contains a code for the synthesis of a protein? No, also. And it determines the order of bases in the protein, which is a function of a tRNA, and it is not the function of that. But ribosome, it assembles amino acid in a chain in a process known as protein synthesis. Now this one, speckled chickens have white feathers and black feathers. This is known as speckled chicken, okay? Have what? White feathers and black feathers. It's telling you here when a chicken with only white feathers and a chicken with only black feathers are crossed, all of the offsprings are speckled. Basically, now let us understand. White feathers and a chicken with only black feathers are crossed, all the offsprings are speckled. Meaning that here I am facing what? Of course, I'm not facing a recessive or a dominant. I'm facing a heterozygous, what? A heterozygous case. So like just the white, let it will be A here and A here, and the black feathers, let just let B and B, okay? Stating here what would be the expected phenotypic ratio if a speckled chicken which contains both the A and the B was crossed with a chicken with only white feathers. White feathers only. Okay, so white feathers only, if you match it together, basically it is what? It is a homozygous was a homozygous. So it is basically 1 to 1. Okay, a speckled chicken will be homozygous. You're gonna get a speckled chicken A and B, okay? And the other one, which is only white feathers, it will be AA. So basically it's a homozygous case. It is a monohybrid inheritance, okay? Just this is a concept of monohybrid inheritance. Just do the crossing and you get the answer. What are stem cells? Basically, they are unspecialized cells that divide by mitosis to produce daughter cells. And this question is repeated a lot in the multiple choice examination papers. And we have, like, if you just solved even paper 4 questions, if you memorize the meaning, you will find it. And it's found in the syllabus content, by the way. Now, still in question 34, what are features of hydrophyte which allow the leaf that float? Float meaning that my leaf, now just let us draw it now. There is many stomata on the upper surface of the leaf. Now I have an upper surface of what? Of the leaf. Now an upper surface of what? Of the leaf. So here is my leaf, okay? Here's what? Here is my what? Here is my leaf. Okay, here's what? Here is my leaf. Now on the upper surface, which is this upper surface, I have more stomata. So I have what? I have more stomata in here. Stomata, which is in the blue, okay? Stomata here, stomata there, stomata everywhere, okay? Basically meaning that this more stomata will help the plant to float. But on the lower surface, which is this one in red, color, okay? This one in red, just let me mark it. Yep, this one. It will what? It will have what? It will have fewer stomata, like 1, 2, 3 or 4 by max. But the upper surface will have more. It's just, the count is just for illustration. It's not real, of course. Of course, the upper surface will have more and much, but it's just for you to understand. Which unit could be used for measurement needed to construct a pyramid of energy? Like just looking into your notes, pyramid of energy, of course, it's kilojoules per meter square per year. You need like per year. Of course, it's not per meter square, because meter square you're not measuring like a land or a farm. It's incorrect. Now per kilogram, of course, not kilogram, because you had already said kilojoules. We are not here measuring the mass of the animals. We are measuring what? We are measuring the kilojoules per meter square of animals per a whole year. In one whole year, how much is the consumption of energy? Question number 36. Which process needs microorganism to break down the proteins to produce ammonium ions? Decomposition. And I just here did it for you here. So basically, we had learned before that decomposition versus denatrification. Just looking into your notes, decomposition produces ammonium ions, but denatrification is a totally different story. Denatrification produces nitrogen, which was first ammonium ions, which is like very totally different case. And in here in that question had made it very clear for you. It's telling you what? To produce ammonium ions, not nitrogen. So basically the answer is decomposition. Nitrification and nitrogen fixation is totally different, and you will be in your carbon cycle, just go, and mean nitrogen cycle, just go and check it and you find it. But the answer for this question, it's letter A, and I made it clear with this comparison in here. Now moving on to question number 37. What is not shown in such a graph? So as we can see here, I have first my lag phase, my log or exponential phase, and I have my stationary phase. What is really missing is the des phase. Des phase. So in here in your des phase, it is missing here. So just we need to refer to the des phase, and it is not shown in our graph, so we need just to complete it for you just to understand. So it was not shown actually. Now, what is an advantage of a large-scale monoculture? Monoculture, of course, you are growing the same type of crop in a larger farm, in a larger place. So this is meaning that you had increased your crop, what? You had increased your crop yield, okay? Why not increase biodiversity? Because they are only the same. Biodiversity means a number of different species living and interacting. Interacting is an ecosystem, but biodiversity is a number of different species. Well, in monoculture, they are the same species. At a larger scale, also you are increasing the same species, so impossible for A to be the correct answer. Increase susceptibility to disease? No, it does not increase susceptibility to disease because it does not have anything to do with the disease. Well, if you have more variation, well, if one plant get disease, it will transfer that disease by cross-pollination or by self-pollination to another plant, and that plant will get sick and that plant will die, and so on and so forth. And increased variation in crop plant, of course no, because in variation we need what does not need a monoculture. We need another types of different species and different plants. So basically the answer here final will be letter B. Question 39. This question has like tripped a lot of students and it was tricky for them, but I don't see anything tricky. You just use my way. Now, stating you the graph show the global emissions of carbon dioxide and methane from 1990 to 2015. Stating a student make four statements about carbon dioxide and methane. First of all, before answering these types of question, I just want you to give an advice. In answering such questions, you need to look into the graph. Observe the graph itself. I just don't want you like to get your information from your knowledge. No, just forget what you had learned. Focus into the graph. It's telling you here, carbon dioxide and methane were only the source of greenhouse gases produced in 1990. So looking into this, if I use my knowledge, I will tell no. Of course, it's not like in 1990. They were only the sources in the graph, right? But in my information, of course, they were not only the source. Carbon dioxide emission continue or rise to the present day. Who knows? To the present day is not like does 2015 is your present day. Well, present day was in this exam. It was October, November 2025. It was year 2025. Like 10 years away from 2015. So basically, it will be incorrect. Carbon dioxide has a greater effect on climate change than methane. Did in the question he told you this information? Did he told you effect or emissions? Did he told you effect or emissions? As you can see here, he told you what? He told you emissions, not effect. He's telling you emissions. Now, methane emissions were lower than carbon dioxide emissions between 1990 and 2015. Yup, methane emissions were lower. As you can see here, this methane, it was lower than the carbon dioxide. As you can see here, like for example, if this one is 60,000 and this one is 70,000, which is already lower than 21,000 or 24,000, which is really like very small. And 1990, 1995, 2000, 2005, 2010, 2015, the methane was like little, very small, very small emissions, okay? Very little emissions compared to the carbon dioxide, which was exceeding the 10,000s in numbers, okay? Now, 40. Which structure is cut to form sticky ends during genetic modification? Well, for that question I had explained it in the previous video for variant 1 November 2025, and also the other previous video I have uploaded on my channel. Just check them. And the answer will be plasmid. Why plasmid? Because as you know, we use a restriction enzyme to first extract your DNA of a bacteria. Just use that restriction endonuclease enzyme to also cut the plasmid, forming sticky ends. So you're just gonna insert the gene with the plasmid, which was joined by the ligase enzyme, forming your recombinant DNA, which was inserted back into your bacteria. I just had like summarized for you the whole story, but forming sticky ends during genetic modification is letter C, which is the plasmids. Which is what? Which is the plasmid to form sticky plasmid. Remember, sticky ends is sticky plasmid. Sticky ends is sticky plasmid. Okay? So that was all about the October-November 2025 paper 2 variant 2 question paper. I hope you had enjoyed. Thank you for your time and meet you in another video. Just don't forget to subscribe, like and share it to your friends so that everyone can benefit, and meet you in another new video.